Okay. We're moving along with the brain, and we're going to look at the coverings of the brain today, and then we'll hopefully finish the central nervous system up and get started on the next chapter. This is figure 22, and a look at the meninges, which are layers that protect the brain and spinal cord on the outside. We have three meningeal layers. The one closest on the brain, directly on the gray matter here in this picture, is the pia mater. Where's the label? There, pia mater. Mater means mother. Some of these anatomical names are a little fanciful. And this is the very delicate covering directly on the brain. The next one out from that is the arachnoid mater, or just arachnoid. And then the outer one is the dura mater. Dura means tough. Arachnoid refers to spiders. And this is because the arachnoid has little spider web-like posts that separated off the surface, off the pia. So there's a gap. The gap is called the subarachnoid space. Subarachnoid space. And in the subarachnoid space, we have cerebrospinal fluid, just like we do inside the ventricles of the brain. And this cerebrospinal fluid in the subarachnoid space protects your brain by forming a hydrostatic cushion around it, so it's not going to be able to move around in your skull very much. So it's not jiggling around in there. So I put a layer of fluid in the subarachnoid space. And then the dura mater is quite tough and leathery. It's a good protection that way, and it's very close to the periosteum of the bone that forms your skull. And you'll see this in lab if you've, well, when's your lab on this? Have you had the brain lab yet? So you'll see this during the brain lab, how leathery it is, okay? So it's a nice protective layer. Now, what's also in this picture are these things. In the midline of the brain, you have a venous sinus right here, superior sagittal sinus, and the arachnoid actually projects through small openings into the superior sagittal sinus, and these projections are called arachnoid villi. Villus is the singular form, arachnoid villi, and this is a mechanism for returning cerebrospinal fluid to the general circulation. We'll talk about where that stuff comes from next. But here's a nice illustration of these arachnoid villi. So cerebrospinal fluid from the subarachnoid space can enter through these villi into the venous part of the blood circulatory system. Before we look at where cerebrospinal fluid comes from, we'll look at this picture here, 23, showing how the meninges, and in particular the dura, actually projects into the deep crevices between the two halves of the brain. So that's this one. So each half of the cerebrum is on either side of that, so that gives a nice stability towards lateral displacement, that the brain doesn't shift laterally. This is called the falx cerebri. And then we have, for the cerebellum, a smaller one, the falx cerebelli, so that the cerebellum doesn't shift side to side. And then we have this tentorium, which is basically separating the cerebellum from the forebrain. And all of those keep the brain from jostling around inside the skull too much. Now, you all know that this only works so much if you have strong blows to the head. You can get a concussion, which is when the system kind of fails, the forces are too strong, and the brain does kind of get compressed into the wall of the brain case, into the wall of the skull. But under under normal circumstances, this keeps your brain from moving around up in there. So you need quite a bit of impact to squish the brain up towards one side. And that causes a concussion because the neurons get compressed and damaged. Now, this is a diagram that shows where the cerebrospinal fluid is actually being produced. This is figure 24. The cerebrospinal fluid is produced in 3 areas that are called choroid plexus. Here's the name, choroid plexus. There's one in the roof of the 4th ventricle right below the cerebellum. This is the cerebellum right here. A choroid plexus right there. And then there's a choroid plexus in each of the lateral ventricles of the forebrain. The choroid plexi are small capillaries that project into the ventricles, and fluid is moving out of those capillaries into the ventricle space. And so this fluid is closely related to the liquid components of your blood plasma. So it's fairly similar to blood plasma. It doesn't have blood cells in it, and it doesn't have some of the large proteins in it that you would get in the blood. But overall, it's fairly similar to blood plasma. It's created by these choroid plexus areas. So we have 2 in the lateral ventricles and 1 in the 4th ventricle. And then this cerebrospinal fluid fills all the ventricle spaces and circulates through there because of the ependymal cells lining the ventricles are ciliated, so they keep this stuff moving throughout the ventricular spaces. And then how does it get to the subarachnoid space? Well, there are a couple of areas where it can escape from the ventricles. One is right here in the brainstem. There's an aperture that's in the midline, and there are 2 lateral ones a little bit higher up in the brainstem, and cerebrospinal fluid can escape into the subarachnoid space from the ventricles. And then it moves through the subarachnoid space around the brain, around the spinal cord, and the arachnoid villi then return it to the blood circulation. So cerebrospinal fluid is continuously being manufactured by the choroid plexi and removed through these arachnoid villi back into the regular circulatory system. So here, the choroid plexus of each ventricle, in this case, produces cerebrospinal fluid. Cerebrospinal fluid flows through the ventricles into the subarachnoid space via apertures. Cerebrospinal fluid flows through the subarachnoid space and then is absorbed into the dural venous sinuses by the arachnoid villi. So we have a continuous circulation of cerebrospinal fluid. And yes?
What's the function of the cerebrospinal?
Well, a lot of it is just protection, physical protection from a hydrostatic cushion. So you have your brain, you have a layer of basically water around it, and then the skull. Then the brain is not in contact with the skull directly, and it's not going to slam into the skull because the water, it doesn't compress really. Liquids don't really compress, so it's a nice protective cushion. And that's its primary function really, and it also has some nutritive functions to the brain cells that are right in contact with it. But mostly the purpose is physical protection. And the next thing we'll talk about now is the spinal cord, which is really continuous with the brain down here with the brain stem. It's also surrounded by meninges. It has the subarachnoid space with cerebrospinal fluid, and even the ventricles continue into the spinal cord as the central canal, and there's cerebrospinal fluid in that as well. So here is a picture of the whole spinal cord. It's protected inside the vertebrae. When you stack all the vertebrae over top of each other, the vertebral canal, that big hole in each vertebrae, they all line up and form that canal, and that's where we find the spinal cords. The spinal cord has several things on it that we need to talk about. One is the fact that it doesn't actually, as the cord, extend all the way into the sacrum. The spinal cord itself ends right there in a little cone shape, about at the level of the first or second lumbar vertebra, so it doesn't go all the way down. This has to do probably with the growth of the spinal cord stopping before the growth of the whole baby stops, so your vertebral column grows longer than your spinal cord for a longer period of time. And from the spinal cord, we have at every gap between vertebrae, we have spinal nerves that exit the cord and go into the periphery. You can see these little yellow things coming off, so those are nerves, spinal nerves from the neck, thoracic, and lumbar region. And in the lumbar region, since there's no more spinal cord itself, all of this is just filled with spinal nerves. And because it's just nerves that sort of grow down, this kind of looks like a horsetail a little bit to anatomists that started describing that. So this is called the cauda equina. Cauda means tail, equina means horse. So the horse's tail hanging down from the end of the actual spinal cord. And this is an area where generally it's much safer to do a puncture if you need to take cerebrospinal fluid. If you do a spinal tap, you usually do it down there because there's much less chance for nerve damage. You're not going to poke any spinal cord itself. You may poke into a nerve, but not the spinal cord if you make a mistake. So that's usually where that's taken from. So regular intervals for the spinal nerves. Now, you see that there are two areas where the spinal cord is actually thicker. Here in the neck region, cervical enlargement, and then in the very terminal part of the cord, the lumbar enlargement. This relates to the fact that the nerve cells that run all the muscles in your arm reside in the spinal cord right there. So you have quite a few muscles in your arms and hands. All of them need to be innervated by motor neurons, and the cell bodies of those motor neurons sit in this region of the spinal cord. And then for the trunk, you have far fewer muscles in the trunk. There's no fine control, so it takes far less of space to pack in the motor neurons for the trunk muscles. And then when you get to the legs, in the lumbar region, then again you need more motor output to run the different muscles in your legs. There's just more different muscles down there. And that's when you have this lumbar enlargement. Here you see a different view of a cross-section, how the cord sits inside the vertebral canal, and it's quite padded, so the spinal cord itself is smaller than the space inside the vertebra. What you see here very nicely is that the meningeal layers all continue onto the spinal cord, so you have a pia right on the cord and the arachnoid with the subarachnoid space, cerebrospinal fluid in there, and then you have a dura surrounding all that. So that's a direct continuation of the brain that makes perfect sense, since the brain and spinal cord all develop kind of at the same time from that rolling in of the surface. You form that neural tube, and the front edge just elaborates into the brain, so it's all continuous. And then in addition, on the outside, then you have packing of fat, structural fat that is there to just make everything immobile pretty much inside the canal in the vertebra. What this also shows nicely is two spinal nerves coming out. So between each two sets of vertebrae, there are spinal nerves coming out at every interval, to the right and to the left. And we'll talk about those in a little while in the next chapter in the peripheral nervous system. Here's yet another view of the spinal cord, sort of dissected out of the vertebrae now. And here they show the outer meningeal layer, the dura, then they take that off, you see the arachnoid, and you take that off, you see everything exposed, because the pia is again right on the nerve cord itself. What's interesting in the spinal cord is that you have kind of a reversal of the position of gray matter versus white matter. In the forebrain, where you do your thinking, you have the cerebral cortex, the gray matter, it's all on the surface, and the white matter, the axon tracts are deep. For the most part, we did have some gray matter deep inside the cerebral hemispheres, those basal nuclei and so on, but generally the gray matter is on the outside, the white matter on the inside, and that reverses in the spinal cord. In the spinal cord, you have the gray matter deep, and then the white matter axonal tracts are on the periphery, so you get that reversal right there. So the gray matter is centrally located. The central canal is pretty small, but it is there, and it has cerebrospinal fluid in it. We'll talk about this gray matter a little bit. You see how it has different areas that are highlighted here. There's a dorsal horn, a lateral horn, and the ventral horn, and two sides connect with a commissure connection right there. And these dorsal, lateral, and ventral horns have very distinct population of cells, nerve cells in it. That we'll look at in the next slide. And then these white matter areas are tracts of axons. So if you look at that on the slide, you see just axons. And they go up and down the spinal cord for communication from the brain down through the cord, and then eventually out through the spinal nerve, and input also. So if you touch something, that touch sensation gets conveyed to the spinal cord, and then from there up towards the brain, so you become consciously aware of what you're touching. If you touched a cactus or a pillow, right? So there's interpretation happening in the brain, and all that information is sent up and down the spinal cord through these white matter areas. You don't have to know these names, but just sort of white matter or white columns, axon tracts. Now for the gray matter, we do need to focus in on what's going on there, and here's a more diagrammatic view, figure 29, off the slice through the spinal cord. And we'll look first here at these gray matter areas. This is the dorsal horn, the lateral, and the ventral horn. And dorsal, again, is towards the back, right, facing backwards. And the dorsal horn has nerve cell bodies that are all involved with the conveyance of sensory information. Okay, nerve cells involved with sensory information. And you see that it's subdivided again into two types of sensory information. We have SS, stands for somatic sensory neuron. Input goes there. This is all the touch receptors on your skin going to that somatic sensory area. So if something touches you on your arm, there's that signal is conveyed to this part of the gray matter in the spinal cord, somatic sensory. The green is visceral sensory. This is input from your internal systems. Input, let's say if you have something in your stomach, that your stomach is turning, there's some input from that. From the visceral organs to internal organs. Any sensory input from them goes ultimately to this visceral sensory area. And then the lateral and ventral horns are for motor neurons, for motor output. The visceral motor here in yellow is motor neurons that are part of the autonomic nervous system that go to your inner organs. Motor input such as stimulating the stomach to churn or elevating the heart rate, something like that. So visceral motor output to stimulate your internal organs going through smooth muscle pretty much and glands. And then the largest region is this red one, this is the somatic motor region in the ventral horn. Those are the nerve cell bodies of the neurons that innervate all your voluntary muscles, your skeletal muscles, okay? So if I'm wiggling my finger, that is triggered by motor neurons in the ventral horn of my spinal cord up here in that cervical enlargement area. So you have this very much stratified inside the spinal cord where these neurons are and what their functions are. And then that gray matter communicates with the periphery through the spinal nerves, and we're going to look at that here right now, and then we'll repeat it when we look at it again in the next chapter. Here is the spinal nerve in the periphery, and you see how each spinal nerve has two branches, so to speak, to it. These branches are called roots. And this one is the dorsal root, and that's the ventral root. Dorsal root, ventral root. And they come together after a centimeter or so in the spinal nerve. So this is the spinal nerve. It has a dorsal root and a ventral root. Now, those roots are just as compartmentalized as the spinal cord itself. The dorsal root is entirely sensory in nature. Dorsal root, here they say dorsal root, sensory. It's entirely sensory. If you were to cut the dorsal root, any sensory input that would go through that dorsal root you would no longer become aware of. You would not feel any sensory input from nerve cells that go through that dorsal root. The ventral root is entirely for motor output from the spinal cord, so here, ventral root motor. So the motor neurons that go to your viscera or to your skeletal muscle sit in your spinal cord, and then they send a very long axon from here into the spinal nerve and then ultimately to your muscles that do this. Okay? So my somatic motor neuron sitting up there in my spinal cord, it sends the action potentials in an axon that leaves the spinal cord through a ventral root and then heads down in nerves all the way to those finger muscles. Okay? So that's why these cells are so long. Big cells. Sensory input. If I were to prick my finger or touch something hot, that comes back through similar nerve endings. There's a very, very long nerve extension, and the cell that actually has the cell body for that sensory input is sitting up in the dorsal sensory root in a swelling called a ganglion. This is the dorsal root ganglion. Remember our definition that we wrote down a little while back? A nucleus in the central nervous system is a collection of nerve cell bodies in the brain or spinal cord. A ganglion is a collection of nerve cell bodies in the periphery, not directly inside the spinal cord or brain. So this is a ganglion. It's a collection of nerve cell bodies. In this case, the dorsal root ganglion is a collection of sensory nerve cells. So these sensory nerve cells are of a particular type that has a specialized axon that actually runs from the periphery to the cell body and then continues on into the dorsal horn. And that's kind of unusual type of nerve cell. So nerve cells in the dorsal root ganglion are entirely sensory neurons that collect information from the periphery and then pass it on to these guys in the dorsal horn. So dorsal root, sensory, ventral root, motor output. Okay, so there was a pretty good start on some of the peripheral nervous system stuff. But now we'll move into chapter 13, and we'll look a little bit more at the peripheral nervous system. So we're done with the CNS, the brain, and spinal cord. Now we'll look at the peripheral nervous system, sensory input, afferents, and motor output, and we have these two divisions, somatic versus autonomic motor output. And in the periphery, most of the axons from the nerve cells don't run as individual axons. Individual axons are pretty fragile. So whenever possible, they tend to run as bundles of axons, and those are the things you would commonly refer to as nerves. if you do gross anatomical studies in med school, you'll learn about the routes of various nerves, and they're never individual axons, they're always like groups, a whole bunch of them together. Like the sciatic nerve, for example, would be a whole bunch of axons that all head down your leg to innervate all kinds of different muscles in your feet and legs. Nerves are pretty tough. If you do dissection of animals or cadaver lab, they're really, really tough. They don't really separate. And that is from lots of connective tissue that bundles them together. So similar to what we had for the terminology with the muscles, where you had individual cells bundled into fascicles, fascicles bundled into whole muscles. Here you have individual axons with a little bit of wrapping bundled into fascicles of axons, bundled into whole big nerves, and lots of connective tissue makes them fairly tough. So they don't separate too much. And the terminology is the same as for muscles. So endo, peri, and epi. For the muscle was mysium was the ending, or mysium, and here is neurium. But it's the same stuff. It's connective tissue. Collagen in particular to make it nice and tough. So what you normally call a nerve is more than one axon running together. Okay. Now, in every class like this, they'll make you memorize some of the major nerves of the nervous system, and we will memorize all the nerves that actually attach to the brain, and then we'll skip the ones that are further down coming off the spinal cords for time reasons. But we are going to do the cranial nerves. Why are they called cranial nerves? Well, those are nerves that directly attach to the brain. Okay, and in vertebrates, we have all the cranial nerves in pairs, and in mammals we have 12 pairs of cranial nerves, and they're all illustrated here, and they have numbers associated with them. The numbers are always Roman numerals, so that's important to remember. And the numbered nerves kind of go from front to back. So cranial nerve number 1 is at the front of the brain, and 12 is in the brain stem. So it's sort of a general rule. And you'll have to remember the names and numbers of these cranial nerves for lab also, and what their basic functions are. So we're going to go over them here, and we will start with cranial nerve number 1, which is the olfactory nerve. And the olfactory nerves are a little bit unusual because that's your one example where it doesn't really form a big bundle of nerves, because we have a very, very short distance to go. This is all going to be from that table 13.2, all these pictures. The olfactory nerve endings have a short distance to go. They go from the inside of your nose, where your sense of smell receptors are located, the olfactory epithelium. And then, if you remember, in the skull, you had to note the cribriform plate of the ethmoid with those holes. That's where they head through. So the epithelium, the sensory receptors are right in your nose, and they just send very short extensions to a part of the brain that's called the olfactory bulb. So this is already part of the brain. So cranial nerve number 1 are just these little nerve fibers going that teeny distance through the cribriform plate and connect to the olfactory bulb. The olfactory nerve has the Roman numeral 1, and it's a purely sensory nerve, so it only has that function of conveying smell input to the brain, so you know what you're smelling, what is in the environment. So function purely sensory, carry afferent impulses for the sense of smell, and damage to the ethmoid bone can disrupt your sense of smells. If you have accidents, car accidents where you hit your head on the steering column, that tends to result in those types of damage. So olfactory is sensory only. The next one is cranial nerve number 2, is the optic nerve. That is also purely sensory in nature. The optic nerve, this is for the sense of vision. Okay? So each eye has an optic nerve that runs back towards the brain, and the optic nerves enter the brain at the optic chiasma, where some of the information crosses over. If you take A&P2, you hear more about that a little bit. And then that sensory information ultimately gets relayed to the occipital area of the cortex. So function purely sensory, carry impulses for vision, optic. Cranial nerve number 3 is the oculomotor nerve. And there's a mnemonic in the book that will help you with memorizing these in order. So that's really the first step. Write them all down, take the first letter, and make some sort of sentence out of that. I don't remember what they use. And then you just have to sort of train yourself to rattle them off. And it's very common, in lab, if you have to name a particular one, you just run through all of them in your brain, or write them down on the margins of your paper, and then you always know which number there are. I forget what the saying is, but there's one in the book that they suggest. You could Google for that too, mnemonics for cranial nerves. There's some really fun ones, sexual ones. So that usually makes people remember them. So olfactory, optic, oculomotor, number 3. As the name already tells you, this is a motor nerve, so this has motor output only. And this motor output goes to some of these muscles that move your eyeball around. So oculo, eye, motor, moving your eyeballs. So it's chiefly a motor nerve, oculomotor, motor to the eye, and it will go to 4 of the 6 ocular muscles. Medial rectus, superior rectus, inferior oblique, inferior rectus. So a lot of these muscles are innervated by the oculomotor. Now you see, if you read the text on here, and this is going to be true for all of the motor nerves going to skeletal muscles, that there's also what are called proprioceptive fibers. Proprioception, this is sensory feedback from the muscles themselves. We've not talked about that yet, but muscles all have little sense organs in them that tell your central nervous system how contracted a muscle is. And all of the motor nerves will have sensory feedback from those muscles that they innervate, but these sensory nerve endings are excluded in determining if a cranial nerve is considered a motor nerve or a sensory nerve. So this one is considered a pure motor nerve, even though it's assumed that, yes, we have these feedbacks from the muscles running through them. Let me see if I can find that where it talks about this. Here it talks about the motor fibers going out, which muscles they go. Here, sensory afferents, which run from the same 4 eye muscles to the midbrain. But this is ignored in determining if a cranial nerve is a motor nerve or a sensory nerve, or as we'll see examples where they have both, and then they're called mixed nerves. Okay? So oculomotor, we consider the motor nerve to those four eye muscles for moving your eyeball around. Cranial nerve 4, right here, is called the trochlear nerve. The trochlear nerve is also a motor nerve. The trochlear nerve also goes to the eye muscles. This one only goes to the superior oblique. The trochlear nerve only to the superior oblique. And it's a motor nerve, but it has those proprioceptors also. So any motor nerve will have those in there, but we ignore them for these functional descriptions. So supply somatic motor fibers to extrinsic eye muscle, the superior oblique. The next one is the trigeminal, number 5. Trigeminal. This is actually a very important one. It's a large nerve that has 3 major branches, and that's kind of what gives it the name, 3 geminal, 3 branches. And this is what is called a mixed nerve. This means it has sensory as well as motor nerve endings running through it. We have these 3 divisions. The print is really small: ophthalmic, maxillary, and mandibular. So this is V. This V is the Roman numeral 5 actually. So 1, 2, and 3. So trigeminal part 1, 2, and 3. Ophthalmic, maxillary, and mandibular divisions. And you can kind of look at a diagram like this here. The ophthalmic division goes to the eye area, so to the upper face. The maxillary kind of to the upper jaw, the maxilla, and then the mandibular reaches down to the lower jaw, the mandible. So we have these 3 main divisions. Here you see what that would look like in a dissection, kind of where they reach. Okay, and each division has particular functions. And the ophthalmic is a sensory division only. Sensory impulses from skin of anterior skull, upper eyelid, nose, nasal cavity, cornea, and lacrimal gland. So all of this upper area, sensory input. The maxillary division, also sensory in nature, from this middle part, from the maxilla, so from that part of the face, nasal cavity, palate, upper teeth, cheek, upper lip, lower eyelid. So this strip right there, sensory input. And then the mandibular division is mixed, so it has sensory input from that lower strip from the mandible. So sensory impulse from anterior tongue, lower teeth, chin, temporal region of skull, so that strip we saw in the other picture. And this has also a motor component to some of the muscles that you use for chewing. So motor fibers to muscles of mastication, that means chewing. So this is the only branch that actually has motor input into the periphery, and that's why the whole nerve is considered mixed. So sensory, sensory, mixed. The whole thing is mixed. Trigeminal, 3 branches. Here you see that again. So sensory, sensory, mixed. So the sensory from this part of the face, and then here are some of the chewing muscles that it innervates. So it goes to the muscles of mastication for chewing. Cranial nerve 6, abducens. Abducens, cranial nerve 6. This is a motor nerve, yet another one to the extrinsic ocular muscles. This one goes to the lateral rectus only. And it's a motor nerve only. So we have 3 cranial nerves for moving your eyes around. A lot of dedication for that fine control. Oculomotor, trochlear, and abducens are all for moving eyes. And they're all motor nerves. The next one is another big one, the facial nerve, 7. The facial is also a mixed nerve. This actually has 5 major branches. You don't have to know those, just the ones from the trigeminal. So this one has even more, and it's a mixed one. Here's an illustration. It does exactly what the name implies. It goes to the muscles of facial expression, so all sort of, if you make faces, that's all facial nerve input. It's fairly superficial. So because the facial muscles are superficial, so this is a major danger for people who have surgery on the face, that these might get accidentally damaged or severed, and then you lose control of that part of the face, which is very disfigured. You wouldn't think so maybe, but our facial muscles really need all that input. Otherwise they just hang limp, and then your whole face sags, and you can't really regain that, since nerves can't be stitched back together very easily. Here it shows you like 5 different branches, but you don't have to know these ones. Mixed input, so you have motor output to all of those muscles of facial expression, and we have sensory input from the tongue. Let me see, where does it say that? Yeah, I think it's in previous. Oh yeah, there. Sensory impulses from taste buds of the tongue, the front part of the tongue, so that makes it a mixed nerve. Otherwise, mostly the motor impulses to face muscles, not for chewing. That was the other one, that was the trigeminal. This is for the face, the more superficial facial expression. There's some autonomic motor outputs to tear glands and salivary glands too. So facial, think about muscles of facial expression, sensory from the tongue, taste buds. Next one, vestibulocochlear. Vestibulocochlear. This is entirely sensory. So this is cranial nerve number 8, vestibulocochlear. Sensory for the ear, hearing as well as balance. This is sort of a two-part nerve. The vestibular part is sensory input from the organs of balance. Over here, semicircular canals. The cochlear part is input from the sense of hearing. And in the ear, there's actually 2 separate nerves that come together. So there's a vestibular nerve for balance and the cochlear nerve for hearing. And then they come together as the vestibulocochlear cranial nerve. So sensory input from the ear, purely sensory. Nine is the glossopharyngeal. Glossopharyngeal nerve, cranial nerve 9, further back. Glosso refers to the tongue, pharyngeal to the pharynx, the back of your throat. This is a mixed nerve. You see its fibers, some of the motor nerves to the pharynx for swallowing. Sensory input from the tongue, taste buds further back on the tongue. So this is a mixed nerve. 10 So motor for swallowing, sensory for taste, sort of the main things. If you read the whole thing, you always find that there's more than I talk about, a little bit more detail. For instance, innervate part of the tongue, somatic motor, pharyngeal muscle, blah, 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 swallowing, salivary glands, sensory taste, general sensory touch, pressure, pain from the pharynx, posterior tongue. So if something is stuck back there in your throat and it's really irritating, glossopharyngeal. Next one is the vagus, 10. This is the only cranial nerve that leaves the head area and heads down into the body cavity to go to the internal organs. It's a mixed nerve. The motor part is mostly autonomic nervous system motor neurons that go to these internal organs and tell them to speed up or slow down. So autonomic motor output for the most part. There are a few going to skeletal muscles in the throat for swallowing also, but most of it is autonomic motor, not the somatic motor. And then you have sensory input from those organs as sort of a feedback. The vagus mixed nerve involved mostly with these internal organs, motor output and sensory input from them. And then 11 is accessory nerve. This is a motor nerve to the muscles in your neck, right here. This one actually starts on the upper part of the spinal cord, then runs through the foramen magnum in and then leaves with the vagus nerve through the same opening in the skull, and that's where its name accessory nerve came from, because you saw when you did a dissection you would see the vagus nerve come out here and then this accessory nerve right next to it. That's where the name comes from, but it's really not an accessory to the vagus nerve. It just happens to use the same opening, and it is a motor nerve to these neck muscles. And I think for some reason your book calls this a mixed nerve, but it's not. It's a motor nerve. Hopefully at some point they'll fix it. And then the last one is called the hypoglossal cranial nerve 12. This is another one that they call mixed, but it's a motor nerve to the muscles in the floor of the mouth. Hypo, below, glossal, tongue. So that's where it goes. And it's for moving the tongue around for the most part. So manipulating your tongue during speech and swallowing is the main purpose of the hypoglossal. So last slide for today. Oh, there they are all again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12, I forgot one. Oh, 12. So you can kind of count them off from the front. And this is a good summary for the basic functions. It has all the Roman numeral, it has them all in order. It tells you if it's sensory function or both. It gives you a general sense what kind of sensory input it is. And you see here accessory and hypoglossal. Here they say no sensory, but in the other part they still call them mixed, right? So go with this, not with what's in that big table. There are no more mixed nerves there in the back. So we'll leave it at this. Hopefully this helps you in lab now that I got this done, because you'll do all these nerves in lab.